Another variation of the chromosome walking is chromosome jumping, where we can construct the jumping libraries. So jumping libraries, they are constructed under those situations where the DNA fragment, it is very large, that it may be from 50 to 200 kilobase pairs. So in this case, when the DNA fragment, it is very large, then the gene of interest on that fragment, they may be far apart. So genes may be at different ends. For example, in the construction of jumping libraries, the genomic DNA, it can be digested with a suitable enzyme, and here the NOT1 enzyme, it can be used. NOT1 is an enzyme that can target at rare sites, so that the DNA fragments that are produced, they are very large. And here the larger DNA fragment, they are circularized, so that the genes that are present at the ends, like X gene at one end and Y gene at an other end, they are far apart from each other. If we will circularize the DNA, so those genes that are far apart are at distant locations, they will come close to each other. And then after the circularization of the DNA, the closure sites, again they are digested with restriction enzyme, so that the genes that once located at different locations, they come close with each other, and then those fragments from the closure sites or the junctions, they are cloned further, and such a type of DNAs uh, that are cloned from the closure sites, they can constitute the jumping libraries. So in jumping libraries, again I can repeat that the genes that are present far apart, they can come close to each other, and then after digestion, the target DNA is cloned to an appropriate vector. For example, here this diagram, it indicates one of the example of jumping clone or jumping DNA library construction. Suppose this is a segment of the DNA, or it may represent a chromosome that contain two genes. Suppose here at left side it may be X and another side it may be Y gene that are far apart from each other, and how we can manage that these two genes, they come close to each other. So it can be managed by the circularization of the very large DNA fragment, so that the genes once located far apart, they come close to each other. Then here the DNA, it is digested, and the jumping clone, it is formed here, and this jumping clone, it can be ligated in an appropriate vector, and further this clone can be used for screening. So this is one of the strategy of jumping library construction. Now what are the applications of chromosome walking and chromosome jumping? The application is in the detection of human genetic disorder. For example, these two techniques, like the construction of jumping libraries, it can be used to detect gene for cystus fibrosus in human. Cystus fibrosus जो है, ये different disorder cause करती है, like different organs like kidney, liver, pancreas जो हैं ये target होते हैं, इनमें genetic disorder होता है, और जो genetic disorder है, it is because of autosomal recessive situation, और ये जो genetic disorder है इसको detect किया जा सकता है by using prenatal diagnosis, so that we may have an appropriate idea that this fetus, it is having this disorder. So the gene of this cystus fibrosus, it can be cloned for the detection of this genetic disorder. So this is one of the application where we can use chromosome walking and jumping to detect human genetic disorders.